our red blood cells have in different humans we have different types. You can have an O type blood, an A type blood, a B type blood, or an AB type blood. And this nomenclature has been developed to recognize the antigens that are found on red blood cells. So for the A type of blood, that means the person has the A antigen, and a B type has a B antigen. An O type doesn't have any, has neither A nor B antigen, and the A B type red blood cells have both the A and B antigen. So the interesting thing is that if you have the antigens, you will not make antibodies to the antigens. So someone with AB blood will not have antibodies to bind the A antigen or the B antigen. If someone has O blood, in contrast, they will have antibodies to both the A antigen and the B antigen. If someone has B type blood, they'll make an antibody to the A antigen, and someone with A type blood will have antibodies to the B antigen. The antibody creation against antigens not found in your body seems to be stimulated by different microorganisms. And that's a whole other thing we could lecture about. But for us, our class, we don't need to know the detail. I just want to mention that microbes seem to be playing a role in these antibodies that are reacting to antigens not found in our red blood cells. So, so sometimes people have referred to the type O blood as being the universal donor, because it's basically the O red blood cells don't have the A or B antigen. So people with antibodies against A or B, they won't be attacking red blood cells of type O. And then the AB type has been called the universal recipient because the individuals with AB blood don't have an antibodies to A or B. However, the problem is that you can't. There's no such thing as, for a universal donor and universal recipient for big volumes of blood. That is only true for very small volumes of blood. And the reason is when blood is donated and given from a donor to a recipient, the antibodies could also be in there, right? So basically, if someone has an O-type blood and then they donate to, let's say, the B-type, someone with B-type blood, right? So the person who has anti-A antibodies, they won't be reacting with the O-type red blood cells. However, with the blood from the donor, there will also be some anti-A and anti-B antibodies, and these could actually lead to the destruction of some of the recipient's B type red blood cells, and that's why only very small volumes can be used when you're changing blood types, right? So if a O donor is giving to a B recipient, it can only be a very small volume. So that universal donor and universal recipient are not true for big volumes of blood. They're only true for very small volumes because in the body, those antibodies would be diluted very much so. But for big volume, like a transfusion, you need to have an exact match. There can't be any, we can't worry about these antibodies because they could cause a really big problem in the body if big volumes are transfused. So here's just a cartoon showing some of the red blood cells and platelets that are found in our bodies, right? So we have the granulocytes on the top, the neutrophils, the eosinophils, the basophils. We have the agranular lymphocytes and agranular monocytes. We have the erythrocytes, which are the red blood cells, and the platelets that are these fragments of other cells called megakaryocytes, and they play a role in clotting in damage. Let's say the wall of a blood vessel got damaged, then they would play a role in repairing that damage. So again, the platelets are also called, by the way, thrombocytes, They're the smallest form element. They're actually fragments of really big cells called megakaryocytes. The platelets lack nuclei. They're very short-lived, living only 5 to 9 days, and they do clot blood with other chemicals and fibrinogen. And there's hundreds of thousands of these per cubic millimeter of blood in the body. So one of the most important processes in our body is the formation of red blood cells. And the process to create red blood cells is called erythropoiesis. And the red bone marrow in our body produces about 2.5 million red blood cells per second. So these are extremely important. The hormone erythropoietin, as shown in this diagram, plays a key role in facilitating the differentiation of these precursors of erythrocytes, and it stimulates the process of erythropoiesis. And it takes about 3 days for this process to occur from the stem cell shown on the top to create the erythrocytes. And also, since the red blood cells are being recycled in our bodies, iron from them, from old red blood cell that's being retired, is usually recycled, and the rest of that, and then sent back for making new red blood cells. But there's also iron from the diet that we get through our digestive system. Our intestine intestinal epithelial cells can import iron through ferroportin channels. And then once it gets across the intestinal epithelial cells, it gets to the basolateral side and gets to capillaries, then it will be carried by transferrin in the capillaries. And one of the important regulators in the body for iron homeostasis is the hormone hepcidin. And what hepcidin does is it removes ferroportin channels to promote calcium storage. To promote iron storage in cells and to lower the levels of iron found in the blood plasma. So again, I just want to start with this process for erythropoiesis, where the stem cell and it'll differentiate, and erythropoietin will facilitate this differentiation process. You do not need to memorize all the little cell types in between. You just need to know stem cell, intermediates, and then it forms a erythrocyte. And the other important thing to know is that the differentiation process occurs in the bone marrow. So that's also critical. So red blood cells are derived from the bone marrow. It takes about 3 days for a stem cell to produce a mature erythrocyte. So the next topic is the structure of the heart. So the heart has 4 chambers. And I want to show you the 4 chambers, and I also want to draw your attention to arteries and veins. So the veins are shown in blue, carrying deoxygenated blood back to the heart. The arteries are shown in red, bringing oxygenated blood, and this is for the systemic arteries in the body. So I want to show you. So here's the inferior vena cava, the superior vena cava, which are veins. They're taking oxygenated blood from tissues and sending it back to the heart. It'll enter the right atrium of the heart, which is this segment I'm showing right now. And this receives the deoxygenated blood from the body. From there, the blood will be pumped into the right ventricle. And the right ventricle's job is to pump the deoxygenated blood from the body into the lungs. And we see it going in here and here. Now, I want to point out something interesting. So even though the blood is deoxygenated, it is leaving the heart and going to the lungs, and we call this a pulmonary artery. Anytime a blood vessel is leaving the heart, we call it an artery. And the pulmonary artery is actually carrying deoxygenated blood. I guess it's very different than the veins, right? The veins usually carry in the systemic circulation the deoxygenated blood, but the pulmonary artery is the one carrying the deoxygenated blood to the lungs. And then there'll be exchange of carbon dioxide leaving and of oxygen being picked up by the red blood cells. And then it enters the pulmonary vein. And again, this is a vein, but it has now oxygenated blood. It's a vein because it's going from a tissue back to the heart. And this is the only vein with oxygenated blood. It goes to the left atrium. From there, it gets pumped to the right ventricle. So it goes to the left. It goes to the left atrium, and then it goes. So it goes from the pulmonary vein to the left atrium. From the left atrium, it goes to the left ventricle, and then from the left ventricle, it goes to the aorta for circulation. And this is now circulating oxygenated blood throughout the body. So I want to summarize the four parts of the heart. We have the right atrium that receives deoxygenated blood from tissues. Then we have the right ventricle that sends the deoxygenated blood into the pulmonary arteries, into the lungs, each of the two lungs. Then we have the pulmonary veins that send back the oxygenated blood from each lung into the left atrium. This then gets pumped to the left ventricle, and then from here it goes to the aorta and to be circulating the oxygenated blood throughout the body. So I just want to point out some important features of the heart. So there's a very fibrous septum separating these different compartments of the heart, and a fibrous septum separates the aorta from the ventricles, the atria from the ventricles, basically. So then, if the atria and ventricles are well separated, the atria can function as one unit for contraction, and the two ventricles can function as another unit for contraction. And the septum also has non-conductive tissue, and that also helps to insulate the two the different compartments, right? The atria from the ventricles, because when the atria are told to contract, the ventricles don't contract at the same time. The atria contract at one point, and the ventricles contract at another point.